So in this video, we will continue with the evolution unit and continue with the concept of microevolution. We're going to be looking at a few just kind of hodgepodge of ideas in this video. So first, we're going to look at this idea of phenotypic polymorphisms. It's kind of a fun word, but it's already something that you understand. Sometimes these are called discrete characters, or another way of thinking about them is either or characters, meaning you either have it or you don't. You either have 100% or you have 0%. There's no in between. When we talk about the next thing, that will make a little more sense. Phenotypic, of course, means your physical trait. Polymorphism means versions of that physical trait. So this is something like witch's peak, or widow's peak, some other people call it, which is what we typically call it in this part of the country anyway. Widow's peak is the little V on the forehead of your hair, and you either have it or you don't. There's no almost widow's peaks or really big widow's peaks or anything like that. You either have it or you don't. There's no degree to that. Blood types are another one. You don't have 70% type A. You have type A. Whether or not you are type AA or type AO, remember O is recessive and doesn't express itself, whereas A is dominant, always expresses itself, that's still type A. And so these are characteristics. This is, when we think about characters, this is typically what we think about. The next thing is called genotypic polymorphism. And genotypic polymorphism is more of this idea of quantitative characters, meaning that a trait there may be several different phenotypic outcomes based on the cumulative effects of several genes. So let me explain. Skin color is a great example of this. We talked about polygenic inheritance and how that had to do with skin color. Skin color is how many dominant alleles do you inherit from your parents, basically. If skin color is represented by, 21 genes or whatever it is, you get a certain number of dominant characteristics from that. And basically that dominant characteristic means that you are producing a certain amount of pigment or a certain protein that produces a certain amount of pigment. If you have 0 of those pigments, meaning no dominant alleles at all, that is called albinism, no pigmentation at all. If you have all the pigments, I don't remember what that trait's called, but there's something else for it. It's this idea that you have all of them. But most of us exist in that continuum somewhere in between. And so this idea of genetic polymorphisms is quantitative characteristics. These are things that add up over time. Now, another way that we can measure variation is on the cellular level. And so if you want to think about, before we talk about sickle cell here, which we've talked about a lot already, you can think of gene diversity. The average human, 86% of the genes in our body are in the homozygous condition. And so only a small percentage are in that heterozygous condition. So there's not a lot of genetic diversity relative, meaning that we all either have the thing or we don't, whatever that is. Heterozygous does exist, obviously, but it's not as big. And then you get down to the smaller differences like nucleotide diversity or even single nucleotide polymorphism, where just the change in a single nucleotide can make a difference in a phenotype. Sickle cell anemia is an example of that. When you get down to looking at nucleotide in diversity, each one of us, as you look at other people, you see a lot of differences, but in reality, we are 99.9% similar to one another as far as our genetic makeup. It's that 0.1% that makes all the difference. And it's pretty incredible to think about. Now, that 0.1% is still a big number of nucleotides, considering we have about 6 billion per cell. And so it's still a big issue. So let's look at some other things that can cause variation. One of these things has to do with geographic variation. These are just differences that can exist depending upon the type of environment that you're in. We've talked about clines before. Cline is just a fancy word for level. You think about incline or decline. And this has to do with how a species can differ depending on your latitude that you're in, or depending on elevation. Elevation and latitude often mimic one another. And so this picture shows this idea that plants at lower elevation typically grow taller than those at higher elevation, which should make sense to us because higher elevation, the air is typically thinner, there's less nutrients or less air, so they're getting less oxygen, they're getting less carbon dioxide, and so they're going to be smaller plants. Should make sense to us. And so that variation does exist depending on those kinds of things as well. Now, evolution has a particular flow to it, meaning that if you were to look at a population and to look at those phenotypic differences or genotypic differences, they're going to exhibit this kind of curve. This is a standard bell curve, which you're going to have one extreme going to be very small percentage of the population, or frequency of individuals. So very small over here, very small over here. Most of the population is going to exist somewhere in the middle. Well, as certain traits begin to get selected for, that flow of evolution is going to change. And there are typically three different flows that can be recognized. In the bottom left, you have directional selection. And this is when that curve moves to the right or to the left. So look at these mice, and maybe not so dark mice are the norm, but then something changes in the environment to where dark mice are going to be more selected for now. Maybe there's a volcano and it covers the ground in dark rock. So the darker mice are going to be less likely to get eaten, whereas the really light mice are just going to disappear because they shine to the hawks above. And so that's what that directional selection represents. One extreme is being selected for, the other against. Diversifying selection is where both extremes are selected for, meaning that sometimes this is called disruptive selection. So you think about the curve as being disrupted, that bell curve. The either extreme can be selected for. There's several different scenarios which this might be the case. I can think of one situation where there's two species, or there's a species of butterfly that one extreme on the phenotype or the other extreme phenotype represents a mimic of a harmful species, and typically birds will leave them alone. But when there's that mixed condition where it doesn't look like either one of the extremes, the birds will eat it. So that would be an example of that disruptive selection. And then stabilizing selection is the opposite of that. That's where the middle is selected for and the extremes are selected against. Great example of stabilizing selection is birth weight in people. Long before hospitals and prenatal care and being able to, understand birth and understand what type of baby you were giving birth to beforehand, so doctors could prepare, people just had to deal with whatever they got. And so if you gave birth to a really small baby, the chance of that baby surviving were really slim. Or if you gave birth to a really large, abnormally large baby, the chance of the baby, or the mother for that matter, surviving was really slim. And so typically the average birth weight was what was selected for. And that'd be an example of that stabilizing selection. So a little bit about this idea of heterozygous advantage. We've talked about this before, and this is where the heterozygous condition is particularly selected for, as opposed to the homozygous dominant or the homozygous recessive condition. Sickle cell anemia, again, is what is going to be the topic here. And this is the topic and this is what I see most of the time. And so what happens here is the heterozygous condition, well, the homozygous recessive condition would be sickle cell, and that is a particularly bad disorder in that it causes some circulation issues and oxygen issues, and just there's a lot of medical issues that can go on with that. But it does provide immunity to malaria. The homozygous dominant condition, obviously they're healthy as far as circulation is concerned, but in these parts of the world where malaria is a problem, the homozygous dominant condition is not immune to malaria. But it turns out that the heterozygous condition has resistance to malaria, but also has a healthy phenotype when it comes to circulation. And so this is where the heterozygous trait then is particularly selected for. They stand out as being the most frequent type of allele that is present in that population. And so you can see here where malaria is, where there's the most malaria, is also where you're going to find the most sickle cell, which makes sense because that particular allele is going to be selected for in those populations. Now let's look at the idea of sexual dimorphism. This is the idea that between the two sexes of any species, there's going to be different characteristics. And so dimorphism means literally two shapes. And so you think of a lion. Lion's a great example. The male is really big and has this, big head and all that fur and the mane. And the female is usually smaller, but has other traits that make the female better. They're usually really good at hunting. The male is just good at, I don't know what really, going around and spraying trees and fighting, probably. That's about it. So this is called sexual dimorphism, where there is a distinct difference between the male and the female of a species. And this creates some issues. So there can be this concept of intrasexual competition. So intra means within. Now, every year, this is one of the most mixed up things, and I don't understand. So intra means within, and so intra within the sexual, so like within males. This is males competing with males. So think of the idea of two male bears fighting for the female's affection. The winner gets the prize, the loser gets to go and lick his wounds. And so this is between the same sex. Intersexual competition is between two different, so it's between a male and a female. So this is the idea where the male or the female, depending on the species, has to kind of impress the other sex. And so like the male peacock showing its feathers and saying, look at my feathers. Or, birds are typically, you see a lot of birds doing this, but not necessarily just birds, but it's try to impress the other individual with some kind of dance or show or some sort of characteristic that the other one sees as particularly fit. And so what would you expect with this sort of thing? If the female peacock says, wow, those tail feathers are really cool, then what would you expect to see in male tail feathers over the course of generations? Them to just get a whole lot cooler over time. And so this is what you typically see in situations like this. And if you go back to this one, what would you expect to see in male bears? They're going to be big, they're going to be strong, and they're going to be able to fight. The ones that don't get to reproduce. And so sexual dimorphism in polar bears is extreme. Some of the males can be like twice as big as the female, and a female polar bear is a really big bear. Male polar bears are like the largest carnivore on Earth. They're just gigantic. And so This sexual dimorphism plays quite a bit of role into the changing of species over time.